its enzymes, which are going to be needed to penetrate and get to the egg. Keep in mind, when the egg gets ovulated, it's not just the egg, okay? It's, you can kind of see it here a little bit. When the egg gets ovulated, there's going to be some follicular cells that are still surrounding it. Okay? They call that the corona radiata, or the, it means like radiating sun or something like that. Anyway, there's still cells surrounding it that have come from the follicle. The sperm have to eat their way through those cells. There's also a glycoprotein layer that's been secreted, known as the zona pellucida. It has to get that through both of those things before it can actually even get to the egg to fertilize it. So that's why you need those enzymes to help out. All right. Fertilization, also known as conception, usually in the outer 1/3 of the uterine tube. Why is that important? Why should it occur in that spot? What does it have to do with?
Isn't it like the timing that it has to?
Exactly. It's all about the timing, okay? It's going to take about 5 days to get from this area into the uterus, okay? And by that time, the zygote will have developed enough to the point that it's ready to implant inside the wall of the uterus, inside the endometrium. If fertilization were to occur more proximally inside of the fallopian tube, by the time it reached the point where it's ready to implant, it may already be down like as far as the cervix, and that wouldn't be a good thing. Okay, so it's all about the timing. That's why it should be occurring out in that outer 1/3. So somewhere around either in the infundibulum or maybe within the ampulla. Thermotaxis, that's just the concept that sperm are attracted to temperature. Okay? They move toward warmer temperatures. So the temperature inside the uterine tubes is warmer, say, than inside the uterus or inside the vagina. So that's one attractant, if you will, for the sperm. The other thing is something called chemotaxis. Okay? The ovum actually releases chemicals. Think of it like perfume. Come this way. You know, it's like releasing these chemicals that trap the sperm, right? You could apply this to like real life with men and women. They get trapped. The sperm get trapped with all these chemical attractants, right? And so that draws them there. Two things, chemicals then and the heat are both moving the sperm toward where the egg is going to be. There's another part in this course where we'll see chemotaxis that doesn't have anything to do with the reproductive system. Anyone know what system chemotaxis is important for?
Lymphatic?
Yeah, exactly, the lymphatic system. What do you mean by that?
Like the immune system?
Exactly. What do you mean by that? Give me more. Keep going. Where are the chemicals coming from, and what are being attracted? Anyone know?
Yeah, I got nothing.
The chemicals are when you have like an infection. Okay, either the bacteria themselves, or sometimes the cells in the infected tissue release chemoattractants. They release chemicals, and those chemicals go out to your white blood cells that are in your bloodstream, and it attracts some of the white blood cells. It causes them to leave your bloodstream, to pass through the walls of your little blood vessels, and then move toward where that infection is. Okay? So very important in helping fight off bacteria, etc. Acrosome reaction, like I said, that's occurring inside the female reproductive tract. It's causing the acrosome basically to weaken and then eventually release its enzymes that are going to allow the sperm to eat through the zona pellucida and the corona radiata. Okay. Again, I think I told you before, does anyone remember sort of the magic number of sperm you have to have in semen in order to be considered fertile for a man?
4?
Right here, 4, 5,000. We're all over the map. Any other guesses?
25.
25. I like the 25. Is it just 25?
No,
25 million?
25 million per milliliter. All right. And the reason for that magic number seems to be that if you have that number, that will mean that eventually about 100 sperm will reach the egg. And about 100 sperm seems to be the number that you need to have enough combined enzymes from the acrosome to actually eat through the corona radiata and eat through the zona pellucida. Okay? Only one of the sperm, assuming everything runs normally, only one of the sperm is eventually going to get through all those layers and get into the egg. And once that sperm actually fertilizes the egg, it triggers changes in the membrane of the egg that basically render it impermeable to other sperm coming in. Okay? But to just get through those outer layers in the first place, you need about 100 or so sperm to provide enough enzyme to get through. Does that make sense? Just say it does.
Hold on, question.
Yeah.
You said that in order for when we have twins, you have to produce 2 eggs.
But that's one way. I didn't say it has to go that way.
Oh,
Yeah,
well,
that's one
because
possibility.
Say, so
Yeah,
it's
because
woman's
you have-
Yeah. No,
DNA or gene.
Because you have identical.
Yeah, but you'll see. There's basically 2 types of twins that you have, right? Identical and then like fraternal twins, and I'll talk about that in a second. Actually, yeah, I'll talk about it in a second. When one spermatozoan enters the ovum, again, the ovum stops collecting sperm. Anyone know what it's called when more than one sperm actually tries to fertilize the egg, when more than one sperm gets through? It's called, what's the prefix in medicine meaning many?
Poly?
Poly. It's called polyspermy. Right? Kind of a fun word to say. Polyspermy. Anyway, that's the name for it. Sperm releases its nuclear chromosomes, a little bit of protein, a little bit of RNA, but most of the organelles, everything else, are coming from the egg. Okay? The egg's a lot larger than the sperm is. Does anyone remember, too, what's the consequence there in terms of mitochondria? What can you find inside mitochondria? Anyone remember from AP1?
The mother's DNA.
Yeah, you find DNA inside of mitochondria. So the implication there is you always get your DNA from your mom. Okay, your mitochondria are always coming from your mom. So the DNA that you have will match up to your mom. So it's a way of following sort of maternal inheritance. I can't remember. Was it this class I talked about like busting a criminal with mitochondrial DNA?
Yeah.
Okay. 20 chromosomes the sperm head, it's fine, makes up 46, right? So that's why you have to have the meiosis during spermatogenesis and oogenesis. You've got to get down to that 23 number before things happen because you're going to be back up to 46 by the end. Does anyone know the name for when you have an abnormal number of chromosomes? It's called aneuploidy. Okay, that means you have an abnormal. We'll talk about that more on Thursday. Zygote, as I said, as soon as fertilization occurs, that first cell that you get in the developing organism is called a zygote, and it's going to become all the other cells in the body. It's like the master stem cell. All right, that was sort of that was leading up to the actual development of the embryo. Any questions about that stuff? Okay, all right. So the prenatal period that goes from conception until birth, almost as soon as the zygote is formed, it starts undergoing rapid mitotic divisions, and that's known as cleavage. Okay, so first it cleaves itself into 2 cells, and then those 2 cells cleave into 4, and then the 4 go into 8, and so on and so on. By about day 3, you get a solid ball of cells called a morula, and you'll get to see some of those in class, I think, on Thursday. We have some models. By about day 5, now keep in mind, as this is happening, as these divisions are happening, the zygote is moving its way down the fallopian tube. So this is all, it's moving and it's dividing. It's moving and it's dividing. By about day 5, that morula has hollowed itself out on the inside. And at that point, it's called a blastocyst. And it's the blastocyst stage that you want to have showing up into the uterine cavity. It's the blastocyst then that starts implanting, working its way into the endometrium. It starts at about day 7, and it's fully implanted by about day 10. Okay? These are, I don't ask a lot of numbers in this class, but I think you should probably know these numbers. You know, I don't go, you know, I'm not going to ask you like how much does the pancreas weigh. I don't, you know, I go into things like that. But some of these things, you should have a concept of like the timing on this early development. All right. If you look at a blastocyst, there's 2 main parts to it. There's an outer part called the trophoblast, and then there's an inner part called the inner cell mass. Those are the 2 main parts. We've got models that, again, you'll see on Thursday, so you get to see these 2 parts. The trophoblast is going to give rise to what's called the chorion, and it's the membrane. It's the part of the membrane that comes from the fetus that makes up the placenta. So we'll talk more about that in a second. It's also from the chorion. What hormone do you think is coming from the chorion? Yeah, HCG, human chorionic gonadotropin. Okay? So, and again, that's the hormone that's going to rescue the corpus luteum, stop it from dying so that the corpus luteum can continue to churn out lots and lots of progesterone. Okay. The inner cell mass, on the other hand, this is the part that's going to become basically 3 things. It's going to become 2 cavities, something called the yolk sac and the amniotic cavity. And where those 2 cavities touch each other, it's referred to as the embryonic disc. So here's the yolk sac, here's the amniotic cavity, and where the 2 come together is called the embryonic disc. The yolk sac in us is an early source of blood cells. Okay? Not nearly as important as in some other animals where it provides lots of nutrients. What animals do you see the yolk providing a lot of nutrients? When you think of yolk, what do you think of?
Chickens.
Yeah, chickens and birds and things, okay? Important source of nutrients. In us, not nearly as important, but does seem to be an early source of blood cells. The reason why it's not as important for nutrition in us is that the placenta develops pretty quickly, okay, to provide nutrients for us. Does anyone remember going back a little bit, before the placenta develops, do you remember how you get nutrients to the developing embryo? Yeah, it's from the endometrium, it's from the glands that are there. Does anyone remember what I said? The fluid that's produced is high in what? It's a storage form of glucose.
Glycogen.
Yeah, it's high in glycogen. So in the first few days that it's implanted, before you really get the placenta developing, like, you know, during week 1, you're getting the endometrial glands providing this glycogen-rich fluid to support it. Once the placenta develops, you get more direct nutrients from the maternal blood. So that's yolk sac. Amniotic cavity, that's a fluid-filled sac. That's the big sac that surrounds the baby. It's filled with amniotic fluid. Basically serves as a cushion or a shock-absorbing sac for the baby during development. Okay. You've probably heard of like an amniocentesis. Everyone heard of that procedure before? Where they go in with a needle and they take a little sample of the fluid here. And what can they see inside that fluid?
Isn't it like birth defects or something?
Yeah, okay. They're not really testing the fluid per se. The fluid's mostly water, but what are they looking for inside that fluid? What's going to be inside that fluid? Cells that have sloughed off from the fetus. So there'll be some fetal cells in that fluid, and then they can test those under the microscope and they can look mostly for big genetic defects, like when you have an extra chromosome, something like trisomy 21 or Down syndrome, where it's something big that you can see. Smaller genetic defects, there's not much you can do with.
Yeah.
Embryonic disc, then that's the spot where the 2 meet, and this is really important because the embryonic disc is the part that's actually going to go on to become the baby. It starts out, the embryonic disc starts out as having these sort of 2 layers, but then eventually it develops into 3 layers that we'll talk about in a second. The placenta is the anchoring spot for the fetus to the wall of the uterus, and it's basically an organ that provides a safe way of exchanging nutrients